In the early 1900s, understanding pregnancy was truly a mystery. There were no simple test kits. Women relied on guesswork or old midwives. These signs couldn't confirm pregnancy early, leaving doctors feeling desperate. In Europe, scientists experimented with animal reactions, knowing urine had changed. Around the 1920s, scientists discovered HCG, a key hormone from the placenta. This hormone signaled new life, proving a woman's body nurtured an embryo. Measuring hormones directly with technology didn't exist just yet. So, they injected rats, mice, and rabbits with urine from the women. After hours, they examined the animal's ovaries. These changes indicated pregnancy. It was real progress, confirming pregnancy with accuracy for the first time. The world needed something much faster, cheaper, and also more humane. Somewhere in Africa, a small frog soon would change all of medical history. This frog would leap into laboratories, unexpectedly becoming a true hero. For millennia, humans sought ways to detect pregnancy, a quest spanning ancient civilizations and scientific breakthroughs. Ancient Egyptians, for instance, would urinate on seeds, hoping their sprouting would indicate pregnancy and even predict the baby's sex. Fast forward to the 1920s, when science began to unlock the mysteries of life, leading to a pivotal discovery. Scientists identified human chorionic gonadotropin, or HCG, a hormone unique to pregnant women, marking a significant breakthrough. The first scientific tests involved injecting urine from women into animals like mice and rabbits. If the rabbit died and its ovaries showed changes, it meant pregnancy, though these methods were slow and required animal sacrifice. Then came the revolutionary frog test, utilizing the South African Xenopus frog. Injecting urine from a pregnant woman caused these frogs to lay eggs, providing a clear result without dissection. Today, rapid home tests detect HCG in minutes, a testament to centuries of innovation. This journey from ancient guesswork to modern science highlights humanity's persistent quest for understanding and progress. Scientists discovered urine revealed pregnancy because of the hormone HCG. Testing was slow, so labs looked for a better method. Hudben did his research work in South Africa. He carefully studied Xenopus laevis in detail. Frogs lived in the water and were sensitive to hormones. Hogben injected the extract. The frog then laid eggs. Could it be that the frog would respond to HCG? Hogben tested the urine samples carefully. The frog laid eggs, clearly indicating pregnancy. It was a simple, reliable, reusable test method. A revolutionary, cheaper pregnancy test was created. Labs kept frogs. The idea spread across the globe. Doctors really loved the quick results they got. Hospitals had frogs. Frogs signaled beginnings. Science's alliance. Woman, frog, hormone. Frog became icon. In the 1930s, South Africa offered the medical world an exciting discovery. A frog could indicate pregnancy. The Hogben test, named for Professor Lancelot Hogben, was innovative. It was fast, simple, and kind to animals. Xenopus laevis, the African clawed frog, became a medical hero. Injected with urine from a pregnant woman, the frog laid eggs quickly. Eggs meant HCG was present, indicating pregnancy. After World War II, this test was widely used. Frogs were exported globally. Hospitals cared for them. Scientists even named their frogs affectionately. By the 1940s, the frog test was standard, offering quick, accurate results. Women knew if they were expecting within 24 hours, a revolutionary change. The test provided emotional support, answering questions before home kits existed. Though eventually replaced, the frog test remains a remarkable achievement. From African ponds to labs, the frog rose to fame. By the 1940s, frog pregnancy tests were standard. The result? A fascinating process. Labs held African clawed frogs in aquariums. They were part of scientific practices. Each frog had a tag. Scientists cared for them. These frogs were precious. Urine was collected containing HCG. A scientist drew urine into a syringe. They injected the sample into the frog. The frog went back to its tank. The lab grew quiet. Scientists waited. If pregnant, urine triggered a response. Within hours, the frog laid eggs. Eggs signaled new life. The frog was unharmed and reused. This was the first reusable test. Scientists loved its accuracy. It quickly confirmed pregnancy. Frogs were tested and returned. Technicians grew fond of their frogs. They were partners. Lives changed through their reactions. The frog's response carried emotion. Technology replaced animals. The frog test's days were numbered. The lab belonged to frogs. The fraud pregnancy test rose to fame in the late 1940s and 1950s, known as the Golden Age. The African clawed frog, Xenopus laevis, was vital to medical labs. Hospitals relied on these creatures to detect life's beginnings from South Africa to the United States. Local collectors caught frogs, packing them for shipment overseas. These frogs became a biological export for science and medicine. Frog rooms were normal, with frog keepers caring for the animals. The frog test was foolproof. Eggs after urine injection confirmed pregnancy via HCG. Results were quick, within 12 hours, and frogs were reused. For 3 decades, the frog test was the gold standard, reliable and inexpensive. By the late 1960s, new tests emerged, detecting HCG without animals. For 30 years, the African clawed frog changed how humanity understood conception. Pregnancy testing has evolved from ancient practices to modern kits. From the 1930s to the 1960s, the African clawed frog was used in hospitals for pregnancy testing. Scientists found that urine from a pregnant woman would cause the frog to lay eggs, indicating pregnancy. By the 1960s, machines could detect hormones in human fluids. This led to chemical methods, using antibodies to bind to the HCG hormone. By the early 1970s, a new animal-free test emerged, needing only urine for results. Hospitals switched, and frog tanks were emptied. Scientists simplified these tests for home use, offering privacy. In 1977, home pregnancy test kits were introduced. Women could confirm pregnancy privately without waiting for lab results. These kits became symbols of progress, working quickly and accurately. The African clawed frog's legacy lives on in every modern test kit. The fraud pregnancy test concluded in the 1970s, marking a shift. Many believed the frog's story ended, but its influence significantly evolved over time. Xenopus laevis effectively detected pregnancy with high accuracy. It became a crucial gateway species for scientific advancement. Scientists carefully studied Xenopus eggs under microscopes. The eggs allowed real-time observation of development and growth. The frog became an important model organism in laboratories worldwide. It revealed life's hidden and complex processes at a cellular level. Labs released Xenopus, unfortunately spreading fungus in the wild. This event taught a valuable lesson about unintended consequences. The frog's legacy reminds us of small creatures' important roles in science. From pond to lab, it uniquely connected nature and medicine seamlessly. The test greatly helped develop fertility treatments and diagnostics. Modern tests began with a single frog, a humble beginning. The frog's enduring spirit lives on in research today. It was indeed a pioneer in the field. It shaped science. Its remarkable story still echoes today.